All right, good morning, everyone. Hopefully everyone feels pretty good with their studying. Hopefully everyone felt pretty good with the first 3 tests. I don't know how many scores y'all have gotten back, but hopefully y'all can get those back soon, so that way you can get them out of your mind. Today's review, we're going to go through lecture 8, 9, and 10. That is all we're going to go through. I don't have any practice questions for you all because of the practice test. Also, today, the actual lecture slot will be filled with Dr. Anderson, Dr. Pross, and I. Both Dr. Anderson and Dr. Pross have made more practice questions for you all to do, so I would suggest going to that. My open office hours will be just following the questions that Dr. Anderson and Dr. Pross have, and it'll be in the exact same Teams meeting. So in the same Teams meeting that they're doing their questions in for lecture, I'll be in there, and once they're done, I'll hold my open Q&A. I won't have any questions for you. It'll just be a time for you guys to ask me any last-minute questions or any clarifications. If you do not want to go to that, like always, you're more than welcome to send me an email. I try to respond quickly to all the emails and concisely with any sort of answer. Before we start with the lectures, does anyone have any questions? All right then. So we will start with the first micro lecture. Get it so I can actually see the chat. All right, so our first micro lecture, as you guys should already be used to it because you have your test tomorrow. At this point, he's just kind of giving you an introduction to micro. You do have to know a few species and a few microbes just as examples, but you won't get into real heavy microbiology until Tuesday for block 2. And like I've said before, that's when I'll actually have more study sheets for you all. I actually have them already laid out and everything, so that way I can scan them and put them on Canvas for you. I'll have one that I'm going to give you all on Wednesday. It's just going to be the first lecture, or the first microbiology bacterial lecture, your Tuesday lecture. Do remember that for this test tomorrow, the microbiome lecture does not count. That is going to be for block 2 test. All right, so as always, references objectives. Dr. Anderson's pretty good at giving concise objectives. I know it seems a lot, it seems pretty wordy, but just kind of go through them so that way you can kind of guide your studying if you haven't done so already. These cases, I know some people have been concerned about these cases. These cases are not going to be tested on. The cases are simply just an example that Dr. Anderson uses, so that way he can kind of get you to think about things from a different perspective. You're going to see a lot more cases later on, and cases like this you won't be tested on for this test block. It will be the next test block that you're going to see question stems more similar to this, that you're going to have to essentially just have this question stem here and figure out all the information to answer the questions. But for right now, he just puts them up there just to kind of get you thinking and to show how useful this information might be and how applicable it is to real world scenarios. So you don't have to worry too much about any of these cases. So here, meeting the microbes. So like I said, this is just going to be an overview in block 2. Block 2 is going to be these first 3 bullet points. So everything that you're doing in block 2 is just the first 3 bullet points. And then block 3 are going to be the last 4 bullet points. Mainly just fungi, protozoa, and helminths. You don't do too much with ectoparasites. But prions, viruses, and bacteria, that's going to be what's upcoming on your next test block. Here, this is just a size of reference. You don't have to memorize anything, but just kind of get a general idea of the different sizes here. So you can see E. coli, so a gram-negative bacteria at the bottom, and how that compares in size to our viruses. So our viruses are much, much smaller than anything else that we have record of for micro. You can see you do have a few viruses that are similar in size to bacteria. We have chlamydia here as a reference. We have paramyxo and the pox virus. So they are very similar in size, but after that, the viruses get increasingly smaller here. So that's just an idea for a frame of reference. There shouldn't be any tough questions on this slide, but it's just something to look at and something to think about when you're thinking relation of size of these microbes. So between a virus and a bacteria, you do need to know the major differences, and I've kind of highlighted them here. So a virus is non-living. It's non-living because it requires a host. So it requires a host to be able to even replicate or do anything. The virus isn't going to accomplish any sort of biochemical process without a host cell. A bacteria, on the other hand, it is free living, which means it can do almost all of its normal metabolism without a host. There are some bacteria that do require hosts for basic functions, and these are referred to as intracellular, obligate intracellular bacteria. He gives an example of those two. Make sure you know those two examples. I know it can kind of be confusing, the difference between them for that aspect, but aside from those obligate intracellular, mainly concentrate on the large differences being non-living and living, and its use of the host. So this is just a reference image here to show you some of the differences between a prokaryote and a eukaryote. So a eukaryote being us, or any sort of mammal, any sort of animal, any sort of arthropod, anything like that. And you can see that we have membrane-bound organelles, being your mitochondria, your Golgi, all of these other, the lysosomes, all of these other organelles that you see in a eukaryote. We don't see those in prokaryotes. Also note the difference here between the ribosomes. So here for a eukaryotic, the ribosome, the total combination of both the large and small subunits create an 80S ribosomal subunit. On the other hand, in a prokaryote or a bacterial cell, we're looking at a 70S ribosomal subunit. So this becomes important later on when we talk about the antibiotics. Some other things to note, the cell wall. So there are some eukaryotic cells that have cell walls, but this is a human-based course, so none of our cells are going to have that cell wall. The cell wall is a pretty differentiating factor for them. Then the cell wall being filled with that peptidoglycan. They also have a single circular chromosome, unlike ours, whereas you know from genetics that there's a good amount of chromosomes that we have. We have a lot more genetic material. They have just a singular chromosome, and we're going to talk about this. This is referred to as a nucleoid. And then they have the plasmids. So the plasmids, that's something we're also going to talk about, being a separate piece of genetic material that gives them virulence factors and antibiotic resistance. So we're going to get into all of this. Just kind of have a general idea of the differences between them. Also, you should see the only organelle that they really have in prokaryotes being the ribosomes. So we saw a whole bunch of organelles in the eukaryotic cell in our cells, but we don't see any really in the prokaryote except for ribosomes. So some of the pieces of bacterial structure, this is just the words to go along with the image. The nucleoid, this has no membrane because we don't have membrane-bound organelles. It is one long circular chromosome. So make sure you note that the nucleoid is just one long circular chromosome. Like we said, the plasmids, they are separate of the nucleoid, so they are separate of that long chromosome, but it is genetic material. And that genetic material can either code for virulence factors or antibiotic resistance genes. And we'll get into how that functions and how we can actually start transcribing these genes if they are not part of the chromosome. So then they have the cytosol. This is just the intracellular matrix here, and there's no organelles except for the ribosomes. And you already know what ribosomes do. So here, this is the entire cycle, the life cycle of the bacterial cells. So we have the lag phase when nothing's really occurring. There's no nutrients, nothing's changing. The number of cells isn't changing at all. Then once they get exposed to nutrients, then we have the log phase, this is their growth phase. It is an exponential growth phase, which means that they are growing in the order of 10. This is going to occur when we expose them to any sort of nutrient. So whether they've infected an organism and they can abuse the energy sources that organism has, or we put them on a growing media. So if we have a growing culture media, we have scientists and pharmacists have put in a lot of energy sources for the bacteria in that media so that they can grow, and they will be in this log phase. Once they use all those resources, they then stop growing and they enter the stationary phase. So this is just they have stopped growing because they don't have any more resources or capacity to use those resources. Over time, naturally, just natural occurrence through ecology, because they don't have any more resources, over time the population will end up declining. So it's important to note that this growth curve that we're seeing here is a natural occurrence. This does not include the introduction of antibiotics. It doesn't include any involvement from the immune system. This is just how it's naturally going to occur. So they start off not growing. They will grow in large amounts. Then they will stop growing because they've run out of resources. And then they will die off because they have run out of those same resources. We talk about it later, but antibiotics will typically attack or use their function during this log phase, because our antibiotics that we have affect some sort of metabolic process. And the only time we're seeing these bacterial cells use their metabolism is when they are growing. So the antibiotics are only going to interfere in this log phase. And then Dr. Anderson showed a very good slide with the diagram that we're going to go into as well, showing the different types of interactions that antibiotics have in the log phase. That diagram is literally simply just a zoomed in portion of this image right here. So instead of showing the entire image, they just zoomed into the log phase. So that way you can see how the log phase changes depending on the type of antibiotic. So taxonomy, this is something you all should know. Just saying that we have a genus and a species. So we are going to be going through all of these, way more than just these 4 examples, but we'll go through some different staph and strep species. So it's important to note that you can have Staphylococcus, but you can have multiple different variants with very, very different genetic sequences for those. And you'll see that more in block 2. This definitely make sure you know these examples of these cell shapes. So you have the cocci, which are just round. Then you have bacilli, which are rods, and then spirochetes, which are just a spiral morphology. It's important to know these because these terms are going to be used in question stems, and you need to be able to think in your head what it might be referring to. Also, the cocci are not always purple, and the bacilli are not always red. We're going to get into the gram staining techniques in a little bit that you need to know, but make sure to know that these colors are interchangeable throughout. So the gram stain reaction, you definitely need to know this. So the test does change every year. So the test that I had is going to be different than the test that you all are going to have tomorrow. But I will say this, we had 2 questions from what I can remember. There might have been more, but it was a long time ago for me, and we had a lot of tests in between there. But we did have 2 questions directly involving the gram stain reaction. One of which was about the actual sequence of events in the gram stain reaction, and then one was very similar to the one that I gave you on Friday about if something were to go wrong in the gram stain reaction. So here, if we have a gram positive and a gram negative, with both of them, when we apply the initial stain, the crystal violet, both will stain purple. Now the crystal violet, what it's doing is actually staining the cell wall. So it's staining the peptidoglycan. You then use the iodine, which is known as a mordant. What that does is that seals in that crystal violet into the peptidoglycan. So because we know gram positive have a very thick peptidoglycan wall, the iodine seals the crystal violet very firmly into their cell wall. The gram negative have a very thin peptidoglycan cell wall, so it doesn't seal it as very hard. So then when you go to the next step, you go to the decolorizer, which is normally just alcohol. That decolorizer will wash away any excess free-floating stain. The free-floating stain would be free in the gram negatives because, once again, they have a very thin peptidoglycan cell wall. But in the gram positives, because you use the iodine, that crystal violet stays in that cell wall. So that's going to leave the gram positives always to be purple. Once you go on to the next step, that's your counter stain. So that's your saffron and red. That will stain anything that's available to stain red. So that's going to stain anything. It's not necessarily just going to stain the cell wall. That's going to stain anything, and that's going to stain the free gram negatives that no longer have any sort of stain to them because of the decolorizer. So make sure you understand and make sure you know this sequence of events and what's occurring and why certain stains are doing certain, or why certain chemicals are doing certain things, whether it be the crystal violet is the primary stain, the iodine is the mordant, then the decolorizer for the alcohol, and then saffron and red is the counter stain. And some of those terms will come up later on I think in lecture 10, you'll see that he refers to saffron as the counter stain and things like that. So our big four type of bacteria, these are the large four. They're not the most common, but these are just the ones that we see a lot. Gram-positive cocci, gram-negative cocci, gram-positive rods, and gram-negative rods. I will say this, for the most part, you are going to see more gram-positive cocci and more gram-negative rods. So those are really going to be the two most common ones that we're going to be talking about in this course. Some not so typical bacteria, this is what I was referring to before. So we have some bacteria that don't fall under those four categories, and it's either because, well, it mainly has to do with a lack of gram reaction. So it has to do with a lack of ability to go through the gram staining process. So acid-fast bacteria, they have a very thick, waxy envelope, which doesn't allow the crystal violet to do an initial stain. So they have to use a different technique known as acid-fast staining. Spirochetes, just because of their shape, the gram reaction will not come out as well. And then you have your obligate intracellular bacteria. These are very important to note. So when we were talking about before, bacteria that require a host cell to actually do any sort of metabolic process, these are your two. So chlamydia and rickettsia. And you're going to go into them much more in block 2, into their details, but right now be familiar with the idea of these are your two obligate intracellular bacteria. It's very, very important to note that. They can be used in question stems, and just the name, and just from the name, you'll have to remember it's obligate intracellular, and you'll have to be able to take those few steps to remember the important things about an obligate intracellular bacteria. And then mycoplasma, it's because they don't have any sort of rigid cell wall. And usually what they do with a mycoplasma is they're going to do what's called a negative stain. And the negative stain just stains everything except for the cell, so then you can see it clearer. So there are other tests that, other sorts of growing medias and other tests that are used for these bacteria because the gram stain doesn't work as well. So our cell envelope. So at this point, the cell envelope is one of the most differentiating factors between the eukaryotic and prokaryotic cells that we can use in any sort of testing procedure. Also, it's going to be one of the most useful things for antibiotics. So the cell envelope is anything outside the cytoplasm. So that includes the cell membrane, the peptidoglycan layers, as well as the outer membrane that's involved in only gram negatives. So Michael, I will not be having a Q&A specifically to review the practice exam questions. I will have, like, as I said before, I will have an open Q&A session after today's lecture. It'll be occurring right when the lecture finishes. I will be in the lecture. So right when the lecture finishes, I'm going to take over and I will have an open Q&A. At that point, you guys can ask whatever you want, but I will not be directly going over anything. So then those are the three pieces of our cell envelope: the cell membrane, then the peptidoglycan cell wall, and then the outer membrane, which is only referring to gram negatives. We're going to see some images here. The differences between gram positive and gram negative. These are very important to note, the major differences between this. So in gram positives, we have a large cell wall. We have only an inner membrane. So it's just one solid cell membrane. And then we have also these other moieties on the outside, teichoic acid and lipoteichoic acid. So these you only see in gram positive. So make sure to note that. And it's going to be something that's going to come up later on, and their function that we actually see for lipoteichoic acid and teichoic acid. Then we get into a slightly more confusing gram negative. With the gram negative, we have an outer membrane and an inner membrane. Because we have those two membranes, we have then what's called a periplasmic space. So there's going to be a space between the inner membrane and the cell wall. It is a very thin cell wall, as you can see. The other main thing that we're going to see is the LPS, the lipopolysaccharide. So that's going to be very important when we talk about endotoxins later on. So that's something we only see on gram negatives, just like how LTA and TA, teichoic acid and lipoteichoic acid, are things that we only see in gram positives. So make sure you can recall all of these differences. We will talk about some of the differences even more, and you'll see. So just putting everything that we just said into words here. So the gram positive envelope, thick peptidoglycan layer, also known as a murin layer, TA, LTA, there's no LPS, and there is no outer membrane because we only have one cell membrane. With the gram negative, it's a thin peptidoglycan layer, once again also known as a murin layer, your large periplasmic space, and then we have on the outer membrane, we have those porins that they use for transport, And then LPS, which is going to be an endotoxin. We'll talk more about that now. And we'll talk about it again later on. Yes.
I have a question. So does gram staining have the same effect in bacteria with capsules, or does the capsules not affect the gram staining procedure?
The capsule won't affect the gram staining procedure, but with the capsule will create kind of like a halo around it. You'll see an image later on when he does he'll show you a gram stain of strep, of a specific strain of strep pneumoniae, that you can see the capsule. So the capsule, you'll still have that purple inside if it's a gram positive when you have that capsule, but then you'll have a thin layer around the capsule of purple, of a little bit of crystal violet that's left behind on the outer wall of the capsule. So it doesn't interfere, but the capsule actually helps the gram staining look clearer and much more definitive.
Okay, thank you.
No problem. And I'll point it out when we actually get to the picture of the capsule, and you can kind of see the halo. The picture's a little blurry because it's very hard to actually do these pictures. So Ryan actually just put it in the chat. You can see the capsule around it, and you can see here this is a diplococci, so 2 cocci gram positives next to each other. You can see that they are purple, and you can see that halo around it, that clear halo, and then a thin layer of purple ring around it. That's the edge of the capsule. All right, so LPS, so once again, LPS just being on our gram negatives. LPS is also known as an endotoxin. The main part of LPS that is the endotoxin is the lipid A portion. What that does is it activates these inflammatory mediators. So the inflammatory mediators is everything that we've talked about before with Dr. Pross that induces inflammation. So your alternative complement pathway, TNF, IL-1, and prostaglandins. Prostaglandins, we've kind of mentioned a little bit here, and you'll have an entire block on them in biochem for block 3. Prostaglandins are just activators of inflammation. That is it. They just activate inflammation in your body. They're actually the main target for NSAIDs, so like ibuprofen and Motrin. These, once it does activate inflammation, lipid A, once it activates this inflammation, it can then lead to shock and cardiovascular collapse. So that's how it works as an endotoxin. So the lipid A portion here, just by itself, if we were to cut lipid A off of the core polysaccharide and the repeating O antigen subunits, then we could insert the lipid A into an organic system, any sort of organism, and it would still induce these endotoxic effects. So lipid A is the main portion of LPS that's actually activating this innate immune system. All right, and then here's our structure of peptidoglycan. So with peptidoglycan, we have 2 main subunits. We have NAM, so this first one, so that's the 3-letter term that we use, so that way we don't have to go through the entire terminology. NAM being this one on the left, and then NAG being the one on the right. So as you can see, we have long chains of NAM, NAG, NAM, NAG, NAM, NAG. You can see it's color-coded. The actual building of the peptidoglycan occurs when we have cross-links between different subunits of NAM. So you can see here that we have a cross-link that is set up between this NAM here and this one here. Then we have another one here and here. So this is how we actually build this is how the bacteria build that cell wall, by creating this cross-link. And we'll get into it in a little bit when we talk about antibiotics, on what's actually allowing this cross-linking to occur, and then how this cross-linking can be interrupted. So Jessica, no, you will not have any gram stain pictures on your test. The only pictures you might have might be a bacterial growth curve. That's about it, but you won't have any pictures. You won't have to identify anything from a gram stain. They will give you information regarding results of a gram stain, and you might have to interpret things from that point, but you'll see more of that in block 2. All right, so then some other structures here, just like we were referring to, the capsule, and then we have the endospore. We're going to talk about these separately. We already kind of mentioned the capsule before in previous lectures, how there are some bacteria that can resist phagocytosis because of that capsule. And this is the image that Ryan put up and that we were talking about here. It's a very, very blurry image. It's very hard to get those microscope images and transfer them to computer images unless you're using a fluorescent microscope. And this is an older picture as it is. But you can see here, you can see it's a diplococci. It's got 2 little round grape-like spheres that are bound together. And then we can see the capsule around it. And some other structures that we see, surface structures being a flagella, which is just going to be for motility, and then the pili. The pili are hair-like projections that you're going to see. When you see a pili, you can see it's very different than a flagella. The flagella, usually you're going to get 1 to 2, sometimes more than 2 flagella, but it's usually unlikely. When you see a whole bunch of hair-like structures on the outside, that's going to be your pili. Also, your pili are usually a lot shorter than your flagella, unless the pili is stretching out for attachment. All right. So some of the functions of pili, these are very important to note. Pili are used in attachment. They're also used in secreting toxins. And then the big one, they're used for transferring DNA. This is something we're going to talk about later in conjugation. But here we have 3 different main functions of the pili. Make sure you know these and you know their purpose. So to attach, and that's going to be later when we talk about pathogenesis, that becomes more important. And that attachment will then lead to secretion of certain toxins, and also attachment leading to transfer of bacterial DNA. Here's an image of the pili, so you can see how many more you would see rather than flagella. Flagella, you would see thicker, longer ones. Here you see a whole bunch of really tiny hair-like structures. And this is what they're going to be using for attachment in whatever they might need. All right, so the endospore. So the endospore is just a survival mechanism. It's essentially just like a little kind of like a little case that the bacteria will put in certain contexts, certain genetic information, and it'll put it inside this endospore, and the endospore can survive any sort of harsh environment. So if you had taken general micro back in undergrad, or if you can remember all that way back, there are certain bacteria that do this, bacillus. So bacillus create these endospores, and what you can do is you can expose the bacillus to a very harsh, either nitrogenous environment or a very harsh pH, which would normally kill the bacteria. So it would kill any sort of bacteria, but the bacteria will then release this endospore, so this little like container of genetic material that won't be affected by the harsh environment. And then once you reduce that harsh environment, you put it back to favorable environments, that endospore will open up and release more bacteria, so that way the progeny of that bacteria can live on. So this isn't just this isn't any sort of replication per se. It's just the bacteria kind of putting some certain genetic material into a shielded area to protect it from that environment. So that way then later on, when the environment changes, then the bacteria can come back and go through and continue to replicate and reproduce. The capsule, this is just an outer layer that we were talking about in that picture, and the entire purpose for the capsule is evading phagocytosis. The pili and the flagella we already mentioned. So antibiotics. So antibiotics create what's called selective toxicity. So what that means, that's just the antibiotics targeting certain factors or processes that differ between prokaryotes and eukaryotes. So the idea behind them is we want to give you a medication that will specifically target a process in the bacteria and not have any sort of side effect on you. The majority of side effects that you see with antibiotics come from dysbiosis, so having dysregulation with the microbiome, or someone having an allergy to that. But the main side effects that we see when physicians give patients antibiotics is they see that we've changed that microbiome, we've changed that microflora, which can then have separate side effects. So the antibiotic isn't targeting your cells, it is targeting specific things in the bacterial cells. And we'll get into that. And that's important to note because there are some other medications that we use for bacteria as well as fungi that they're not selective in their toxicity, so they can't be referred to as antibiotics. Antibiotic is something that is selective for the bacteria and for differences between bacteria and eukaryotic cells. So this is that diagram that I was talking about before. This is just a zoomed in version of that log phase. That's all it is. So if we were to expand this chart out even more, you would see the lag phase over here on the left, you see the stationary phase over here, and then you would see the death or decline phase here. So what the antibiotic is actually doing, it's inducing either early death phase or early stationary phase. So our bacteriostatic, they inhibit bacterial growth, so they force the bacteria into an early stationary phase. Bactericidal will induce an early death phase. So they'll skip that stationary phase completely and induce a death phase. Now, these are just induced phases. This technically isn't the actual stationary phase because the stationary phase is when they run out of any sort of energy or resources. but the antibiotic is kind of inducing that stage. It's stopping them from growing any further. And then the cidal is forcing them to die in that time period. So make sure you know the difference between that. Make sure you know this diagram. Also know the examples. So we do have here just penicillin and tetracycline. There are other examples later on when we talk about the different antibiotics. You need to know whether it's bacteriostatic or bactericidal. So some other definitions that we talk about when we're talking about antibiotics, the spectrum, so the range, whether it's a narrow or a broad range. So narrow just being it's focusing on a certain species. Broad could be looking at a certain genus, or it could even be looking at a certain gram reaction. So a broad spectrum a broad spectrum antibiotic could be used against all gram positives. So make sure to note that difference. And then know the MIC, the minimum inhibitory concentration. So this is just the minimum concentration required of the drug to stop growth. So this doesn't refer to bactericidal or bacteriostatic. This isn't saying that this drug, this MIC, is determining that this antibiotic specifically is static or cidal. This is just saying this is how much you need. This is the minimum amount that you need of this antibiotic to stop growth. Just stopping it. It's not saying that it's going into static or it's going into cidal. It's just the overarching term of we are seeing no more growth once you get this amount of bacteria. So here we have a diagram representing that. This is a clear broth that we see here. The broth has certain amounts, certain different concentrations of antibiotic, and then we're going to put in the bacteria. If the broth changes color, that's showing that we have bacterial growth. So here in this first one, 8 micrograms per milliliter, no growth, all the way no growth until the MIC being 1 microgram per milliliter. After 1 microgram per milliliter, then we start to see growth. That means that 1 microgram per milliliter is the minimum amount of concentration you need of antibiotic that stops growth. Once you have less than that minimum amount, you will have growth of bacteria. So make sure you note that and kind of understand that reference. So narrow range and what it affects. So narrow is just it's just a terminology used for the type of antibiotic. So narrow can be an antibiotic that is used for a specific type of species of bacteria, or it can mean any sort of just like a genus. So if we have a whole bunch of bacteria, if you're you're infected and we're not too sure with what you have, we go through a whole bunch of tests. We're growing bacteria on cultures and we can't figure out the exact species, but we know that you have a staph infection or just any type of staph. And we can pick a certain antibiotic that is going to be targeted for staph specifically. It's not going to say Staph aureus, but it's just, or Staph agalacti. It's just this specific antibiotic works towards staph infections. Broad, on the other hand, so like the tetracycline, that's going to be used for anything that's an obligate intracellular bacteria. That's not the only thing it's used for. It's also used for a lot of gram-negative infections, but let's just use the overarching term of obligate intracellular. So if we are doing any sort of culture testing and other tests and we see, okay, we haven't gotten to the point yet where we know what type of bacteria it is, but we know it's an obligate intracellular bacteria, let's administer tetracycline because it covers that entire umbrella of all those bacteria. So that's the difference between them. Is broad is just you're covering all of your bases with a broad antibiotic. With narrow, it's something that you transition to later on as a physician because you have now figured out almost exactly what type of bacteria we're dealing with in the infection. Does that make sense? Hopefully so. All right, so now we're getting into the actual meat of our antibiotics, the different types. So we have 4 major groups, 4 major classes. There are more than this. You'll get into it in pharmacology. You have an entire test on just antibiotics and all of their names and all the different generations. It is a lot. But for right now, you just need these 4 classes of antibiotics. And they're classed through their mode of action. So inhibitors of peptidoglycan synthesis, inhibitors of nucleic acid synthesis, inhibitors of protein synthesis, and then inhibitors of metabolic processes. So we're going to go through all of these and we'll break it down. I also made that pseudo study sheet that I'll bring up. Hopefully you all have seen. It's a pseudo study sheet because I didn't actually make it when I was studying it. I made it while they were going through the lecture, just really quickly off of how I would have made a study sheet. So our first one being the peptidoglycan synthesis inhibitors. So what these do, these are beta-lactams, and they are bactericidal. That should make sense that they would kill the bacteria because they are stopping the bacteria from actually creating its own cell wall. So if it doesn't have its cell wall, then the internal contents would leak out and it would lyse itself. These are your two examples. Make sure you know these examples. Make sure you know it's penicillin, cephalosporin. Now, there are differences between these. Even though they are beta-lactams, they are different generations, and they work slightly different. You don't have to worry about that now, but just know that there is a difference between them, even though they're classified under the same umbrella. So here we can see, once again, our structure of our peptidoglycan. What's actually going to occur is when we are doing cross-linking. When the bacteria is creating this cell wall, it has this long chain of NAM and NAG, and it's going to connect this chain to this chain through NAM-NAM interaction. And that's done by a protein called peptidoglycan binding protein, PBP. So what that does, that protein actually creates this little cross-link right here that I've got the mouse on. That little cross-link right there is created by that peptidoglycan binding protein. It's also known as penicillin binding protein. So here he just put PBP. It's also known as penicillin binding protein because that's what penicillin actually binds to. So penicillin binds to it and inhibits its function. Now you can see here this protein does two things depending on the type of bacteria. With the gram-positive, it's going to bind that lysine residue with the 5 glycines to then the D. I think the D is the glutamic acid. I think. It's been a long time. And then with the gram-negative, you have a different interaction with the penicillin binding protein. So make sure that you note that between the gram-positive and gram-negative, penicillin binding protein is working differently. So this penicillin binding protein, our penicillins just bind to it, and that prevents the cross-linking. So if we can have that beta-lactam, I can tell someone has their mic on. And I'm
Excuse me, Danny, I have a question.
Mm-hmm.
What is it that one residue between the two gram bacteria? Which one is the one residue between them?
So if you look at the picture, you can see it's highlighted in red, and you can see where the cross-link occurs. So here on the gram-positive, it's a L, so a leucine to a lysine, to then glycine, to then D, which I think is glutamic acid, and then alanine. So that's going to be the gram-positive. You just see it in the slide 39. And then in gram-negative, you can see that it's DAP, which is a different protein residue that's binding then to a glutamic acid. So we have a direct bind here with gram-negative, and then with the gram-positive, we have lysine and 5 glycines in the way.
So alanine is common for both of them, right?
The D and the alanine are common in both of them.
Oh, they do. Okay, perfect. Thank you.
No problem. So the way that these...
Is it important that we know that?
I would be familiar with it. I would
Okay.
be very familiar with it. Whether or not... Okay, it's aspartic. Thank you, Ryan. Yeah, it's been forever since I've had to do amino acids, so at least I was right that it's acidic amino acid. It's not necessarily that they might ask you a direct question on it, but it would come up in a question. I know it did for us, and they were specifically referencing gram-positive in that case. So it would just be something to be familiar with. If you are having trouble with the material and you're still trying to get through and trying to churn and burn through it all, I would leave that on the back burner. But if you're very comfortable with the material, then try to remember these things, because that can be the separation between the 92 and the 90 to get you that A. Because as you know, you only have 4 questions that you can miss to retain an A on these tests. So some of these little things, I know they seem minute, and a lot of other people like to throw around the terms high yield and low yield. Me personally, I don't agree with categorizing anything as high or low. I think just know the big overarching ideas. Once you know the big 3,000-foot view ideas, then you can work into these tiny little details that can help differentiate things in test questions. So
Okay, so just to confirm, the lysine, it's binding to the lysine for gram-positive, and for gram-negative binding to the aspartic acid. That's like the main difference?
It's binding to the DAP, the DAP.
Okay.
So you have the binding between the lysine and the glycines and then the aspartic acid, but it's really with penicillin binding protein for gram-positives, it's binding that lysine. And then for gram-negatives, it's binding that DAP region.
Okay, thank you so much.
No problem. All right, so for the actual effect of these beta-lactams, what they do is the beta-lactam enters the cell, and then it binds that penicillin binding protein. So remember, this penicillin binding protein, they can also use the term peptidoglycan binding protein because it's going to do both Mainly, they're going to use penicillin binding protein because it was found, this actual protein was found when they were trying to figure out how penicillin actually worked. So by binding penicillin binding protein, it stops that cross-link. So it stops that mechanism that we see in slide 39. Because we stop that cross-link, we no longer have that cell wall. Because we don't have that cell wall anymore, then we get autolysis of the cell. So the cell just dies itself. Here, this is a very good thing to remember, the central dogma of biology. He has, I believe it's the very next slide, he has which antibiotics work in which section. This is also something I did in my makeshift study guide. It's pretty important to kind of remember it in this way. If you draw it out this way and you add which antibiotics work at which section, you know, which ones work at DNA replication, which ones work in transcription, translation, it can help you remember not only their function, but where they occur, so what they're affecting. So it can help you remember, you know, here quinolones, they work on topoisomerase. Well, if it works on topoisomerase, it's going to inhibit DNA replication. So it kind of add and collect all of your thoughts a little bit and create that process. So here he kind of did it for you, and I kind of did it in the study sheet. I would definitely add a lot of information. For all of these antibiotics, you need to know the general class, what it does, how we see resistance to them, and a few of the examples. So you don't need to know all the examples, you don't need to know all the names, but some of the things that they work for. So I know some of them, like tetracyclines, he mentions that it's very good for obligate intracellular. So make sure you know that kind of idea. You need to know if it's bactericidal or bacteriostatic. So our first ones for our central dogma that inhibit nucleic acid synthesis. So we have two here. We have quinolones and then we have rifampin. Quinolones are bactericidal and they act on DNA gyrase. DNA gyrase is just another name or another form of topoisomerase. So if it's inhibiting topoisomerase, it's not allowing that relaxation of torsional strain. So then you're actually going to destroy the DNA that the bacteria has. So by doing that, then you are eliciting a cidal effect. Know these two names, so norafloxacin and ciprofloxacin. Just be able to remember that the oxacin goes with a quinolone. Just have that in the back of your mind and be prepared for that. Rifampin, this is different. This is going to stop DNA to RNA. So this stops the DNA-dependent RNA polymerase. So that prevents DNA from binding and it prevents transcription. So here with quinolones, that stops any new DNA from being made. Rifampin is going to stop any new RNA from being made. So make sure you can differentiate between those two because they are different. He doesn't say here, but I'm pretty sure rifampin is known as bacteriostatic. So make sure that you have that in the back of your mind. I'm pretty sure I also have it on my study guide, the little makeshift one that we'll go over. So translation. Bacterial translation is very similar to eukaryotic. The biggest difference is the ribosome number. So here we have two subunits, the 30 and the 50S. And these two subunits combine to make a 70S, unlike in ours that are 80S. So it's important to note that because you do need to know the antibiotics and which subunit they attach onto. Here is a very crazy diagram. You don't need to know this. I think this is just way too confusing. I think it's a lot easier if you just write it out and just put which one does which. So the very first one, aminoglycosides. Aminoglycosides bind to the 30S, so that's the smaller subunit. By binding to that, it causes a misread of mRNA. So we already have the mRNA made, but then now as we're going through, the mRNA is being misread. So it's either causing destabilization of the complex while we're ruining the ribosome, or it's causing the bacteria to create improper proteins. By doing that, it will then elicit a cidal response. This is why this one is cidal and rifampin is static. With rifampin, we're just not making mRNA. But here we have the mRNA, but we're misreading it. So we're either creating poorly manufactured proteins, or we're destabilizing the entire complex, which will then cause the bacteria to die. This one, so you can see here, it is a broad spectrum. The newer ones are broad spectrum, and it's mainly only on aerobes. So that's an important factor too, that they don't use it for anaerobes, they only use them on aerobe bacteria. Some of the mechanisms that we see of resistance, so we see 3 here. So the bacteria can either alter the drug. So once the aminoglycoside gets into the bacteria can then change some chemical structures onto it, altering the drug, changing its effectiveness. They can reduce the uptake so it can stop absorption of that bacteria into its cell, or it can mutate its binding proteins. So in this case, it'll mutate, it'll go through and change with genetic variation. It'll change some of the binding proteins on that 30S subunit. So now the aminoglycoside can't bind to that 30S subunit, and it can't prevent that translocation. Then our next one.
Fast. Sorry. What do you mean by reduced uptake? Like reduced uptake of what?
Of the actual antibiotic. So the antibiotic, when we administer it, it's just a chemical. It has to go from your bloodstream into the bacteria. So there are certain glycoprotein receptors on the outside of the bacteria that will grab the antibiotic and bring it in. So it'll just kind of engulf it. It'll go through endocytosis and bring the actual antibiotic inside of its own cell. And that's how the antibiotic will be able to interact with the ribosomes. What they can do is they can prevent those uptake mechanisms. And you'll see that we also have another form of resistance where they allow uptake, but they increase efflux. So they increase the amount that gets pushed out. So it's reducing the effectiveness. So here, this is just the bacteria not taking in the aminoglycoside.
Gotcha. Thank you.
No problem. So tetracyclines, these are a little bit different. These also bind to the 30S, but they bind in a different way. They bind to the 30S to prevent tRNA binding. So that's really the big thing here. So you still have your mRNA, you still have everything going. You've created your ribosomal subunit. You don't have issues with stability between the 30 and the 50S. What you do here is you can't bind your tRNA. And if you remember, tRNA is actually what brings your new amino acid pieces to create your long peptide. So if they can't do that, they can't create peptides. So this one's going to be static. Because it's not directly killing, we're not creating improper proteins, we're not doing anything like that. We're just stopping them from creating proteins, period, because they're not allowing that binding of that tRNA subunit. So those are going to be static. With that, this is going to be used on obligate intracellular. It's going to be used on way more than just obligate intracellular, but when you have an infection that is obligate intracellular, or you assume it is, you're going to use a tetracycline. So tetracyclines can be used on a very broad spectrum. They can be used on a lot of bacteria, mainly on a lot of gram-negative bacteria that you're going to see in the next block. So it is broad spectrum. Make sure you note that. But when you have specifically obligate intracellular, this is the antibiotic that you're going to use. Here, this is the other form of resistance that I was talking about. So efflux. So what this is we have an efflux pump. The bacteria have an efflux pump to push things out of their own cell. What they do is they increase the affinity of that pump to tetracycline. So by administering tetracycline, the tetracycline gets brought into the bacterial cell. But now, while it's in here, before it can activate and bind to that 30S and the tRNA, it has a very strong affinity for that efflux pump. So it gravitates to that efflux pump just kicks it right out. Same thing. As it comes in, it just gets pushed out. So it goes through this whole cycle of being brought in and pushed out that it can't actually work its function. That's just another resistance mechanism, which is why there's more than just one version of tetracycline. You know, we have doxycycline, things like that, that we create, that pharmacists create, so that way when you have an efflux pump that has very, very high affinity for tetracycline, well, now we've created a doxycycline. So it's very similar. It has very similar effect. It can still bind that 30S, but it does not have the affinity for that efflux pump. So it's just trying to stay that one step ahead. And when we see obligate intracellular, should we think yes? So to answer Ryan's question in the chat, and you all can see it because I have it shared on my screen. When you see obligate intracellular, the first thing that should come to mind is chlamydia and rickettsia. So if there's any question in a test that is referring to chlamydia or rickettsia, first thing in your head, obligate intracellular. Okay, it needs the host machinery. What can we use to beat it? We can use tetracycline. So kind of make those connections in your head.
And also that resistance through efflux, it's also another kind of reducing uptake, correct?
It is not. No. So a reducing uptake is stopping the antibiotic from coming into the cell. A resistance through an increase in efflux is pushing more antibiotic out of the cell. So the antibiotic can still go into the cell, but it can't go through its actual function because the moment it gets in, it's kicked out. And then it'll come back in. And then once it comes back in, it gets kicked out again immediately.
Thank you.
No problem. All right, so our macrolides. So our third one for translation. This one is different. This one binds to the 50S. So make sure you note that your aminoglycoside binds to the 30S, causes a misread. Your tetracycline binds to the 30S and the tRNA, and then your macrolide binds to the 50S subunit. So by binding to the 50S subunit, it blocks translocation, period. So that is also going to be bacteriostatic. Once again, we're not inducing cell death. We're not creating any sort of protein or breaking down the cell wall to induce cell death. We're just stopping protein formation. So that's going to be bacteriostatic. So some of the examples of this one, erythromycin, I would be familiar with that. This one originally was a very narrow spectrum bacteria or antibiotic. The new ones that they've come up with now are more broad spectrum. And you've heard of that if you've ever had a Z-Pak before. A Z-Pak is azithromycin. So it's just a newer version of this macrolide. So the newer version is very broad spectrum, which is why anytime you have any sort of sinus infection, earache, anything like that, you go to the doctor and they're like, here, take a Z-Pak. 5 pills later, you feel much better. And this is why. So this one is a drug of choice for mycoplasms and Legionella. Make sure you reference that. And what this does, some of the resistance that we see with this one is modification of our rRNA. So what it actually is, it's modifying, the bacteria is modifying subunits on its big 50S subunit. So it's actually specifically modifying the 23S, which is just part of our 50S, and that's just to stop binding of the macrolide. So that's just to prevent the macrolide from being able to bind to that 50S. And that's why, once again, we have erythromycin as our very first macrolide, but there's multiple different variations of erythromycin that we have, so that way we can avoid these resistance factors. And then our last one, so our metabolic inhibitor. This one is just a combination of 2 antibiotics. So it's not 1, it's just 2. So the question being, why did they switch to a broad spectrum? So sometimes, if you think about it, it's tough because you normally want to use a narrow spectrum. That's the main idea, is you want to be able to target it more. But we have issues when we can't. So if you have a bacterial infection, let's say you have sepsis, you have bacterial infection in your blood, and it is very, very bad. You know, you got to the hospital and it's already a little late. Like, we're seeing a large number of bacteria in your bloodstream, and it's starting to affect your organs. Before we can have a narrow spectrum antibiotic, we need to find out what type of actual bacteria that is. Well, we need antibiotics that we can administer that are broad spectrum before we give you that narrow spectrum one. So that way we can start using an antibiotic that is widespread, and it's going to kind of lower the amount of bacteria that you have in that blood. So it's a first base treatment of like, all right, we're going to give you this now because we know it's some type. It's it's either gram positive or gram negative because we got that from the gram stain. So we're going to give you one that works on gram positives first, just to cover our bases until we can find out exactly what bacteria this is, and then we can change you onto a narrow spectrum. So that's why they come out with those broad spectrum ones. So they have that coverage in those in between periods. So resistance for the macrolide. So for a macrolide, the resistance, the bacteria modifies the 23S RNA subunit. Remember, an rRNA is just your ribosomal RNA. So to make a 50S and a 30S subunits, they use rRNA. So if it modifies the 23S rRNA, it's changing substituents on that. It's changing some of the base pairs. By doing that, it can inhibit the macrolide from binding to that 50S. Because remember, all of these are chemical compounds that have interactions. So if we have, if there is a specific hydroxyl group on the 23S that allows the macrolide to bind, if the bacteria removes that hydroxyl group, now the macrolide can't bind. So if it can't bind, then it can't actually interrupt translocation. So that's what you're going to see with most of the modifications, is a genetic change to change any sort of interaction, and that will inhibit the antibiotic's function. So here with the metabolic inhibitors, they're going to inhibit folic acid. So folic acid, it's going to be something, if you haven't gotten into in biochem yet, it's something that we as humans need, but we don't make, and bacteria actually make. And they use these enzymes, tetrahydrofolate and dihydrofolate reductase. They use these enzymes. So we have 2 different antibiotics that we use here, but we use them in conjunction because together it increases the effectiveness. So we have sulfonamide, which is just a conjugate form here of a precursor. And then we have trimethoprim, which inhibits that enzyme, that tetrahydrofolate reductase enzyme. So by doing that, we put a different subunit in here to prevent this precursor, and then we also inhibit the enzyme that prevents the action of folic acid. So folic acid is going to be used in creating amino acids. It's also going to be used in creating nucleic acids. So if we can inhibit both of those, then we can very, very strongly affect the bacteria. So here, this is just going to be static because once again, it's preventing amino acid formation. It's also preventing new nucleic acid formation. So it's preventing new formation. It's not directly inducing death. They are broad spectrum. So remember, anytime we're talking about this here, we're talking about combining them together. So this is using both antibiotics in this situation. So it is static and it is broad spectrum. Resistance here occurs in two different methods. And this should make sense because we have two different antibiotics. So the first one being resistance for sulfonamides, decreasing permeability. So that's going to decrease the uptake of sulfonamides. That's going to prevent sulfonamide from actually getting into our bacterial cell. And then for trimethoprim, this one's targeting the modification. So once again, this is changing the subunits on tetrahydrofolate reductase. So here, because trimethoprim acts by inhibiting tetrahydrofolate reductase, the enzyme that they need for folic acid synthesis, if the bacteria changes subunits on that enzyme, trimethoprim can no longer bind to that enzyme, and then it will lose its effect. All right, so here is a good study question. You might see something like this. His study questions are very good. They're on point, but they are not the same as the test ones. They are slightly lower level than the test questions. So make sure you can go through these and you can answer them. Like I always say, when you're going through, answer it, but then also go through which ones are wrong and why those answer choices are wrong. That'll help you expand your knowledge a little bit more. And then this is some good basic information that you need to go through.
Sorry, Daniel, for that last question, is it okay if you go over just that one?
This question here on slide 53? Yeah. Okay, let me read it because I haven't read these. Which define best explains the high selective toxicity of penicillin. Okay, so penicillin, immediately when we read this, the first thing we should go back to is what is penicillin's function? So penicillin prevents peptidoglycan cross-linking, right? So penicillin doesn't actually act on our cells. And we should know that because we don't have peptidoglycan, right? So if we don't have peptidoglycan and penicillin only interrupts peptidoglycan synthesis, penicillin will do nothing to us because we don't have peptidoglycan. So the answer should be 2, peptidoglycan is absent from eukaryotic cells. Does that make sense?
Thank you so much.
So here, like, you should be able to go through that process on all of them. So like here, bacteria make their own folic acid. Well, penicillin has nothing to do with folic acid. That's sulfa and trimethoprim. Then bacterial ribosomes function differently than eukaryotic ribosomes. Well, penicillin has nothing to do with ribosomes. The ones that do have to do are aminoglycosides, tetracyclines, and macrolides. And then I would go through, again, what each of those does and how they are affecting the ribosome. So you should be able to kind of create things like that and when you're going through and studying these questions. And some points to know, he puts this at the end. These are overarching points, no more than just this. This is just like bare minimum. You really need to dive into it a little deeper. And then these are very good practice questions if you have the textbook. It's once again, he's not going to use the exact same question you see here for his actual test. He does a pretty good job on making sure that test questions should not be ones that you've seen before. But these are very, very good practice to go through the correct thought processes and kind of filtering out information. So then really quick, I'm going to bring up the study sheet that I did. So this is pretty terribly done, but you know, it is what it is. I kind of made it in like 6 minutes for you all. And yes, my handwriting is that terrible. On future study sheets, I tried to make my handwriting a lot better and clearer because other people were using those study sheets. Because a lot of people ended up actually using my study sheets in the program last year just for micro. So here, if we were to turn that entire lecture into one study sheet, this is what I would have done. I would have put our staining techniques over here on the top left, you know, and put down what they actually do, the different stains, and then what the result will look like. I put the shapes of the bacteria and the bacterial growth curve. In the middle, I would have filled up this entire section with all the information regarding the capsule, the LPS, the differences between positive and negative, the different factors that we see with the nucleoid, with what we see with plasmids, things like that. I would put all of that information in here. And then I would have filled the entire perimeter with the antibiotics. So that's all the information just in one sheet that you can do. And I would suggest writing these. I've suggested writing them from the very, very beginning, because typing it, you don't actually absorb the information. If you go through the process of writing it, it encodes into your memory faster. So you can see here I put the central dogma at the bottom, and I kind of added the information with the quinolones and the rifampin, and then RNA to protein for translation. I put the different types here, and I put a little mnemonic ATM, and it kind of goes in that order. A being aminoglycosides for the 30S misread, T tetracycline for tRNA, and then M macrolide, and it's the last one, so it's the 50S. Also, you definitely put in the what they're used for. So, some of them said broad spectrum, some of them said narrow spectrum, some of them said, like the tetracycline obligate intracellular, things like that. And then also put the resistance factors, and then you can throw in the other two here. So the cell wall synthesis inhibitors, and then the metabolic inhibitors over here. So this is just kind of a general idea. If you haven't looked at this, make sure to look at it. If you haven't created your own, definitely create your own, because this is bare bones. This is not enough to actually study. You need to add a lot more information in this to really prepare yourself. All right, let's go to the next lecture. So our next lecture, genetics and diagnosis of bacterial infections. Okay, going back to peptidoglycan wall, I think the L and D refer to the relative configuration of amino acids rather than the actual. You'd have to double check. I'm pretty sure they mean the actual substituents. I don't think they mean the optical configuration. I think the L and D actually refer to the type of amino acids, but I could be wrong. But that's something y'all could check out on your own. That's something that definitely won't be on the test, is whether or not it's amino acid or it's the optical configuration. All right. So genetics. Here, his objectives again. The reading assignments are pretty helpful, but they're only helpful if you read them before you go through his lectures. So that's something that I would make sure to note, that try to start working ahead on those. Block 2, the reading assignments are very helpful because it gives you a lot of information on the different types of bacteria and viruses that you're going to be going through. So our bacterial genome, we have different locations for our entire genome. The genome isn't like ours where it's just all contained in one long strand of DNA or multiple chromosomes. Here we actually have different locations. We have the nucleoid, like we said before, and in the nucleoid, you do see on average about 4 million base pairs. These numbers aren't concrete. It changes. As you can see, he changes them later on, actually, so I wouldn't worry too much about them. Just get an idea of relative size. So we have one nucleoid per bacterial cell. Plasmids, on the other hand, we have more than one. So remember, plasmids can be for virulence factors or antibiotic resistance. So we're going to see a lot of them in something along the lines of like a MRSA. A very antibiotic resistant bacteria will have a lot of plasmids, and also it's very deadly, so it has a lot of virulence factors. So you're going to see a lot more plasmids in that case. And then pathogenicity islands. These are in both because this is what actually makes the bacteria a pathogen. And we'll go into later what differentiates a pathogen from normal flora. I think that might be in the next lecture. So our genome here, like we said, we have our nucleoid, which is just one long circular double-stranded DNA. Then we have our plasmids. These are independent of the nucleoid, and these code for that antibiotic resistance and virulence factors. Virulence factors just being anything for adhesion or any sort of toxin. So these plasmids or these virulence factors are commonly exchanged between bacteria. This is how we get evolution in the bacteria. This is how we can get one bacteria being pathogenic and the next one, it can then conjugate so that the next one is pathogenic. This is also how we can reduce pathogenicity. This is what they did in his actual example of the BCG vaccine, how they got that Mycobacterium bovis and they grew it over time and they reduced its virulence, was through these processes. So because that virulence can change between, if you grow it long enough without having to use its virulence factors, then that bacteria will die off as a pathogen. And it's the same thing that they do in the lab, so that way they can study E. coli and staph without having to worry about it causing any sort of infection. So here, this is just the lactose operon, the LAC operon. You're going to see this a lot in MCAT studying, if you haven't done that already. And this is also just basic genetics right here. So we have our normal strain right here. A, this is our normal lac operon. In this case, we have the lac operon there, which means we are going to get mRNA. If we have our repressor, so the repressor fits in and moves the operon, then we have no actual mRNA transcript. And then here, slightly different. So this is the repressor, but the repressor has an inducer. So that way it inactivates that repressor, and then we can still get our mRNA transcript. It's just basic genetics. Just be kind of familiar with this example here. So pathogenicity islands, these are a little bit unique. This is something that actually Dr. Blank might have already talked about. If not, he will mention it a little bit, but I think he did. It is present in genomes of pathogenic bacteria. So if it is a pathogenic strain, you'll see pathogenicity islands. If it's not a pathogenic strain, you won't, hence the name. But like we said before, we can get that pathogenicity island to go from one bacteria to another. And that's something that we'll see later on when we actually talk about the types of replication, whether it be transduction or conjugation. Here's your image of it. Something important to note here is with this pathogenicity island here, we do see a lower number of G and C. So we see lower of GC binding. So that's kind of important to note. I kind of mentioned it in passing. So we see that outside of the pathogenicity island, we see about 50% of GC binding, but inside we see a reduced number. So that's a way that they can go through PCR and do gel electrophoresis and find out whether it is pathogenic or not by just looking at that piece of DNA and seeing it's the amount of GC binding. So this is how we can exchange genetic information amongst bacteria. We have transformation, transduction, and conjugation. I tried to highlight like key words here. So transformation is naked DNA. So that's when the actual bacteria is uptaking naked DNA. Transduction is actually inducing that, or it's introducing that DNA through a virus. So the bacterial virus being a bacterial phage. So the bacteriophage actually injects that DNA into the new bacterial cell. And then conjugation is just referred to as sex. It's done through physical contact. We're going to go through each one of these just like Dr. Anderson did. So here, transformation. So in transformation is just uptake of naked DNA to exchange those certain factors, those pathogenic factors. So here in this first example, we have a live encapsulated strep pneumoniae. So the encapsulated is what allows it to be pathogenic because it can avoid the immune response. And it is live, so that way it can function. So this live encapsulated one, we introduce it to a mouse model, then that mouse will die because it is pathogenic because it's encapsulated. If we have a live one that is unencapsulated, so it should not be pathogenic, doesn't have the actual code for that capsule within its genome, so then the mouse's immune system will be able to react and kill off that pneumonia, so then we will not have a dead mouse. The mouse will survive. In this case at the bottom, we have a live unencapsulated, so a non-pathogenic one, and then we have a pathogenic one that is killed. So it's important to note here, this one is killed. So this one cannot go through replication, it cannot go through the infecting process. It is just the DNA there of that organism with the capsule. So if we introduce both of these, normally in this situation, the mouse would survive, but because this bacteria now has access to this free-floating naked DNA of this killed pneumonia that has the DNA for the capsule, that capsule and that pathogenicity island will transfer over to this one that before was not pathogenic, but it now is, which will result in a dead mouse. So that makes sense to everyone here? So we have here a live one that is pathogenic, kills the mouse. A live one that's not pathogenic will not kill the mouse. If we administer both of them and the one that is pathogenic is killed, so it's not going to function, even though this one's not functioning, this one that previously did not have the capsule can take on the DNA and now have the capsule and then go through and kill the mouse. So that's how transformation works. It's just this one here that's unencapsulated was just taking up the DNA of the killed one and incorporating its DNA into itself, so now it's pathogenic. So transformation is naturally occurring, as you can see. Sometimes it can be induced in the lab. This is done for genetic cloning. It's something that y'all would have done in a basic genetics lab when you do it with the fruit flies and making them glow in the dark. That's kind of what you're doing there. This one does require the bacteria to be in the log phase, which should make sense because the only time it would take up DNA from its surroundings and incorporate that DNA would be in a growth phase. So it wouldn't be occurring at the beginning of the growth phase, but it would be occurring in the growth phase as a whole. All right, and then conjugation, that's the second example. This one is just referred to as sex. So the reason it's referred to as sex is because it does require physical contact. So here we have an F+ cell and an F- cell. The F is just referring to the plasmid. So remember, we know what the plasmid is for, antibiotic resistance and virulence factors. So this one, the F+ has the plasmid, and it's going to transfer its plasmid over to this F-. So what it does is it imparts that pili on its surface. This is known as a sex pilus, and it'll adhere both bacteria together. In here, it then creates kind of like a tube to allow genetic material to go from the positive to the negative. When that happens, now we have plasmid in both. So once we have plasmid in both, that pili will be gone. And now we have positive in both bacteria. So before we had one with the plasmid, one without. Now we have plasmid in both. So now both bacteria have virulence factors or antibiotic resistance. So here's just an image of it. So we can see it is one-way exchange. That one-way exchange is going from the positive to the negative. It can only occur from a plasmid, from one that is positive for the plasmid to one that is negative to the plasmid. It wouldn't occur from a positive to positive because there's no need. Also, at the same token, if this one's positive and that one were positive, this one would have pili around it as well. And then if it has that pili, it actually won't need to make interaction with the sex pilus. So make sure you note this one only occurs from an F+ to an F-, and it's a transfer of that plasmid from that positive to that negative.
Wait, Daniel, sorry, the only the F+ has the plasmid, correct?
Yes.
Okay, thank you.
No problem. So here, transduction, the last one, transduction is introducing DNA through a phage. When we say phage, we're talking about a virus. It is a bacterial virus. And there are two different types, either specialized or generalized. And we'll get to a picture example of that, which explains it a little bit better, in my opinion. So bacteriophage is just a virus that infects bacteria. There are two different strains or two different types of bacteriophage. We have a lytic strain and a lysogenic strain. The lytic infects and directly kills the host cell. You're going to see both lytic and lysogenic when you get into normal virology with Dr. Tang. So kind of get a grasp on it now so that way you're prepared for it. Lytic is just going to cause lysis. So here we're not integrating any sort of DNA. We're not going to see transduction occur in the lytic stage. In the lytic stage, we're going to inject the DNA of the bacteriophage or RNA will go through, it'll replicate, and it'll replicate to the point where it's creating so many new bacteriophage within the cell that it'll then burst the host cell. In lysogenic, that way we actually are integrating the DNA. So here, this is when the bacteriophage will infect the host. It'll inject its DNA or its RNA. That RNA or DNA will then incorporate into the host DNA, and it will over time create different bacteriophage. Over time, the lysogenic can then mutate into a lytic. So the lysogenic is when it injects that DNA, the DNA incorporates, and then later on in that cell's life cycle, that lysogenic DNA can then start to replicate, and it will then lyse the cell. So you can go from lysogenic to lytic. You cannot go from lytic to lysogenic, and I'll explain this a little bit more in the picture. So here in this picture, on the left side, this is going to be lytic, and on the right side is going to be lysogenic. So with the lytic, like we said, we have the bacteriophage here, and it's going to introduce its genetic material into the cell. That genetic material will then replicate over and over until we have more bacteriophage. Here it's just showing 3, but essentially you would be getting hundreds and hundreds and hundreds until all of these phage fill up this bacteria, and it's too much, and the actual cell just bursts. And then we release more phage, and then the phage will go on and continue the process with other bacteria. In lysogenic, we can see here that we inject the genetic material. That genetic material will then integrate into the nucleoid. As it does, when the bacteria go through replication, we'll see more bacteria that have that integrated genetic material. Over time, this genetic material will then activate and start creating phage, and then it will lead to lysis. So this pathway goes here. This integration pathway, the lysogenic pathway, will go here, and then it can come right back up into replication and go into lysis. So this one's going to integrate the DNA. This one's not going to integrate it. This one's just going to cause direct death. So make sure you can note those two differences. And here's just another example. This one is going to be the example of specialized and generalized. So specialized, we're only using a small piece of the genome, a very small piece that is special for certain toxins or certain proteins. Generalized is an incorporation of the entire genome of the phage. So that's just the difference between them. In specialized, it's a small piece that encodes for maybe a toxin or a certain receptor on the outside. Here with generalized, this is something that we see where the entire genome is incorporated.
Daniel, I had a question on that. Sorry to interrupt you. With regards to the specialized and the generalized, I see that there's 3 phages, one with just the black, one with the black and the red, and one with the red. Which is specialized and which is generalized?
The names next to them. So the specialized one is going to be the one that has half red, half black, because it's only the red is only the certain portion of the genome. And then the generalized is all red because it's the entire genome is going to be incorporated.
So I'm assuming the one that's all black is also generalized technically?
No, so this would be the preliminary one. So this is the initial one.
Gotcha. Okay, thank you.
No problem. All right, so our transposons, this is something a little different. This is something that can move within the chromosome. So this is sections of our genetic chromosome that can move within a nucleoid. So we can have one piece go from one side of the nucleoid to the other, or it can go from a nucleoid to the plasmid, or from the plasmid to the nucleoid. So this is how we actually get antibiotic resistance. So we said before that the plasmids code for virulence factors and antibiotic resistance. Well, how does the antibiotic resistance and the virulence factors actually go through transcription and translation if they're on the plasmid? Because the plasmid doesn't go through that. The plasmid doesn't go through protein formation. The nucleoid does. And this is how, by them being transposons. So they go from the plasmid to the nucleoid, so to the chromosome, and then there they can be transcribed and they can be translated into proteins to actually activate antibiotic resistance or activate virulence factors. So with the antibiotic resistance, if it goes from the plasmid to the chromosome, then it can then encode on an efflux pump and create higher affinity in that efflux pump for the antibiotic, or to eliminate any sort of entry or uptake, anything like that. So this is what the transposons actually do. So some of that drug resistance, this is an antibiotic resistance sensitivity test. So we get bacteria, we grow it on a plate, and then we put these discs. These discs, each of them contain a different antibiotic, and that antibiotic will permeate through the media and will either kill off the bacteria or it'll show us that the bacteria is resistant. So we can see here at the very bottom on this image, we can see these halos around some of these. So that means that E. coli is sensitive to some of these antibiotics. So it is killing. That halo is the death of the bacteria in that area because of that antibiotic. Then over time, that resistance will develop because it's being exposed to it. We'll talk about that a little bit more here. So this is just an image that he goes through of how we get that transposition, so that transposon going from plasmid to nucleoid. So here's your transposon, here's your nucleoid. If we have here, I believe he had a purple line here that you can't remember, pink, magenta, whatever color that might be. You can see it's a hard line here. This hard line will jump from the nucleoid onto the plasmid. You can see that occurring here. The purple line is gone. We've switched that purple line here. You can see it, or the pink, whatever you might want to call it. And now we replaced it with a different piece of genetic material from the plasmid. So this orange bar might encode for antibiotic resistance or some sort of virulence factor. All right. So here, the development of antibiotic resistance. So he has 3 main ways that he talks about how antibiotic resistance comes about: overprescription, improper usage, and then animal feeds. Overprescription, that's going to be something that we see with like the Z-Pak, like I was talking about before. There are actually a lot of articles coming out now how Z-Pak's azithromycin is losing a lot of its effectiveness. And that's because for a while there, for a good 7 years, anytime you went to the doctor with anything, any sort of sinus infection, any sort of ear infection, anything, they would immediately hand you a Z-Pak. Say, here, take this, take azithromycin, you'll be fine in 5 days. And that works. But as we start exposing bacteria more to these broad spectrum antibiotics, then we're allowing them chance to be exposed to it and then create forms of resistance. Because remember, this resistance doesn't just pop out of nowhere. It's an evolutionary change. It's an evolutionary change to, okay, here's this antibiotic that's killing all of my friends. What can I do to stop that from happening to me? And then once that one bacteria survives with that resistance, then it will go through conjugation, it will go through transformation, and it will replicate more bacteria with that resistance. So that's something that we see a lot with that overprescription. Improper usage is where they refer to when the doctor tells you that you have this antibiotic cycle. It's supposed to be 7 days. You take it for 5 because you start feeling better. Well, now by taking it for 5, you've exposed some of those bacteria to antibiotic, but they haven't died because you haven't gone through the full regimen. So you've killed a majority of them, which is why you feel better, but there's still some stragglers left behind. Those stragglers now have time to create and go through evolution and create a sense of resistance to that bacteria or to that antibiotic. And then animal feeds. Animal feeds is essentially the same thing as overprescription, because we are just all the animals that we have that we are doing to increase crop and increase production. We expose them to these antibiotics to prevent them from having disease so we can have more food. And then by doing that, we're just exposing all of these bacteria to more and more antibiotics. I think someone had a question in the chat.
Yeah, I had a curiosity question. And it left my head that quickly. Oh, the way we do the antibiotic resistance, or the MIC test to see the minimum concentration, how do we develop the guidelines for knowing the time that we need to expose the bacteria to? So where the doctor says take it 5 days or 7 days.
That's going to be
developed?
So that's going to be there. It's going to be a combination of the MIC it's going to be a combination of 3 things: the MIC, this antibiotic resistance test here, and then as well as the assumed quantity in your body. So if we assume, depending on the type of infection that you might have, let's say you have sepsis. With sepsis, because it is more widespread and you have a media that the bacteria can grow on throughout your entire body, with sepsis, you're going to have a longer time span for that antibiotic because you have so much more bacteria to kill. If you have a sinus infection, the sinuses are actually very contained. So you have those few sinuses that you should go over in anatomy soon if you haven't gone through the skull yet. They're very, very contained. So because they're contained, we know that there's a limited quantity of bacteria that can grow there. So we can extrapolate that minimum inhibitory and to the resistance factor to see if it's strongly sensitive or weakly sensitive. Then we can extrapolate that and say, okay, well, the limited amount of bacteria that can grow in the sinuses will be dead with this antibiotic because it is strongly sensitive in 7 days, 5 days, things like that. So they take that out and they go off of how strong the antibiotic is, as well as the minimum inhibitory concentration. If the minimum inhibitory concentration is much lower, that means the antibiotic doesn't have to work for as long. And then they go through where it is, so how much bacteria can actually be there in that area. Does that answer your question?
Yes, thank you.
Okay. All right, so here we can see antibiotic resistance in Staph aureus. This is something that we see. It's called MRSA, and we'll get into that a little bit later, methicillin-resistant Staph aureus. And we can see we have a hospital strain and a practitioner strain. So the practitioner strain is something we see in normal environments, and then so it's the antibiotics working very, very well against it. You can see that almost all the bacteria is completely dead, and we have a lot of these zones of inhibition. With the hospital strain, this one has been exposed to much more patients and a lot more different antibiotics. So because it's living in the hospital, it actually has encountered all of these different antibiotics, and it can have time to build that resistance. And we can see that in this one, that very few antibiotics work with this one. Gene jumping is from plasmid to nucleotide, which then integrates. Yes, to answer the question in the chat for Gabriella. Yes, gene jumping, which is transposition, is certain genes jumping from the plasmid to the nucleoid, and then it incorporates in the genome, and then those genes can then be transcribed. So our super germs, this one, like we were saying, MRSA, this is resistant to a lot of different antibiotics. The reason they say methicillin is because that is the strongest penicillin strain that it's resistant to at the moment. These are also known as nosocomial infections. This is a term that they're going to throw out a decent amount. Nosocomial just means it's hospital acquired. So it's something that you're going to get, you're mainly going to get from the hospital. There are community acquired MRSAs that you can get in assisted living facilities or gyms or things like that. But the ones that we're mainly going to be talking about are nosocomial infections, so hospital acquired. And then identification and diagnosis. We're going to go through a few different examples. There are certain ones that Dr. Anderson pointed out that we're going to go through. So the culture, it's labeled as the gold standard because that is the first thing that is done. So anytime if you work in a hospital lab or in public health lab, the moment you get a specimen, first thing you're going to do is you're going to gram stain it, and then you're going to create cultures. So you're going to either put it on a blood plate, a chocolate plate, a McConkey plate, depending on what bacteria you might be thinking about. The whole purpose of this culture is to identify the bacteria. Then you can do antibiotic sensitivity testing, the plates that we saw before where they actually put the little discs that are soaked with antibiotics on them, and then strain typing. Strain typing is going to be more so genetic testing. So the types of culture media, so we have solid, which are agar plates, as you're going to see most of the time. Liquid media, so there are broths. We can use that in blood cultures, which you see at the hospital when they take blood cultures for any sort of infection. There's also other types. There's a brain heart infusion one to see that allows you to grow in, to mimic cerebral spinal fluid, things like that. So that way you can grow the bacteria in an environment that it would enjoy, so you can isolate it better. Selective and differential media. So selective media is when we want to inhibit bacteria from a culture in a non-sterile environment. So we have certain areas in our body that are non-sterile. So the mouth, the gut, the intestinal tract, any of those regions are listed as non-sterile because we have our normal flora living there. So selective media would inhibit the growth of the normal flora and only allow growth of the pathogenic microbe. So that way we can only grow the pathogenic, because if not, we'd have a plate full of bacteria and we wouldn't be able to figure out what is causing the problem. So selective media is going to inhibit unwanted growth of other bacteria, mainly non-sterile, from non-sterile environments. Differential is where we can differentiate between two separate types of bacteria. So once we have the selective media, so let's say, we have that media, we have stool sample. So the stool sample, we're going to use a selective media, so that way we can get rid of all the non-pathogenic microbes and we only grow the pathogenic ones. Once we have that, let's say we have 2 or 3 on there, then we're going to get those 2 or 3 and plate them onto differential media. The differential media will help us figure out, is this E. coli? Is this a pathogenic E. coli? Or is this something else? Is this a Clostridium? Is this a Bacteroides? What else could be growing here? The differential is going to help us figure out, okay, we have these three, which one is the pathogenic one? And then cell cultures, these are a little bit more rare because these only occur in obligate intracellular bacteria. So we use these when we need to grow those obligate intracellular, because if not, as we know, these obligate intracellular will not grow without a host cell. Morphology, this is something you guys really won't have to know or be tested on, but this is something when you work in a lab, if you work as a pathologist later on, if you go that route, that you kind of get used to, and you can kind of recognize how the bacteria grow and how certain strains grow on certain plates. Dr. Anderson will reference some examples of colony size and shape and appearance in the next block when you start talking about certain bacteria that you will have to be aware of. So just make sure, right now it's not something too pertinent, it's just an overview, but next block you will have to know that. And here's some different examples of selective and differential media. We have blood plates here, we have MSA down here at the right, at the bottom right, and an EMB on the left. You don't have to know these examples. They'll be referenced later on in block 2, but this is just to give you an idea of what they'd look like. And then differential media, this is a McConkey plate. This is one that you can use to help differentiate between E. coli and not. So you can see we have 2 different types of colonies on here. We have a clear one and then the purple one. So we have coliforms and non-coliforms, depending on what type of sugars that they can process. So it's a technique used to differentiate between bacteria, so that way we can kind of find out what's the heart of the problem. Some other tools, the gram staining, the acid-fast staining, immunofluorescence, that's not used as much, and then special stains are only used in certain circumstances. The gold standard here for identification is going to be the gram stain. It's the first thing that they do with any specimen is they run a gram stain. So to do the gram stain takes about 5 minutes. Then after that, you have to let it dry out and then read it. You all should know the procedure at this point. We talked about it. Like I said, it's very important. Here is where you're seeing some of that other terms like the counter stain. And then the cell shapes and size. This is what you see when we're talking about the gram stain. Remember, the colors are important. You're not going to see this color as much in a gram stain. With a spirochete, you will do other ones, but these are very rare to come across. The main ones that you're going to come across are going to be cocci or bacillus, so round or rods. And here's some examples of some gram stains. A and B, these are gram positive. You can see here he's got gram positive rods, bacilli for A, and then B are going to be cocci. And then here we have the same thing, rods on the left, bacilli on the right, and C and D are gram negatives. So you can see the difference in color between them. It's pretty evident. Fluorescent microscope, this is something that, like I said, is really not used that often. It's going to be used more so when you're doing genetic testing. So that way you can actually go through the process and research base. It's not used clinically as much. And then there's other types of identification. So biochemical analysis, these are going to be used a lot. These are used for as differentiating medias. So carbohydrate analysis, so gas production, pH change. That's an example of the McConkey plate, like I was saying before. And then enzymatic, catalase, and coagulase. You're going to go through these in block 2. So you'll see a few more examples of that in block 2. Just be aware that you have these different ways that you can use to identify and differentiate these bacteria. And then you also have genomic testing, so where you're going to use PCR. You guys should know about that, not to bring up any bad memories from Wednesday's test, but be familiar with that process of PCR and kind of keep in the back of your head what it's used for. So we have direct detection of antigen, and we have direct detection of DNA. For direct antigen detection, the main one we're going to talk about is ELISA, and then DNA detection is PCR. The PCR is used a lot. Both ELISA and PCR are used a lot in clinical settings. PCR you're going to come in contact with more frequently. And whenever you go to the hospital, they're going to do a nose swab and a rectal swab if you have to stay in the hospital for an extended period of time. The nose swab is going to be a PCR for MRSA, and the rectal swab is going to be a PCR for VRE, which is another resistant superbug. So the ELISA here, we have antigen detection. We have 2 different ways that that we're going to show how to do the ELISA. One way, we have the solid medium with an antibody on it, and that antibody will then attract specific antigen that we're looking for in the sample. Once that antibody and antigen are bound, we'll wash any excess off, and then we'll apply another antibody with a marker on it. So it can be a fluorescent marker, any sort of marker that can help us know we do have antigen bound. So this antibody is going to bind to the antigen as well, and it'll create that sandwich. So we'll have antibody, antigen, antibody sandwich here, and the top one will have that fluorescent marker to let us know. This allows you to find out if there is antigen in the sample, in that patient sample that you were looking for. And then PCR, you guys should know this. The picture that you saw in genetics would have been pretty similar to this. So just kind of be aware what it is and what's going on in this system. Ivy went through that with you all. Something else we do is we do indirect presence of antibodies. This is looking for a titer. So a titer is when we do serology testing, so serum testing, to look for specific antibodies. It's not always a specific test, because remember, it's not telling you that, you know, we have this strain of IgG that is made for this type of bacteria. It's just telling you we have high levels of IgG. What you can do is you can then take that titer and then use that same serology and use it onto an ELISA. And we're going to talk about that type of ELISA. So we have the direct ELISA, where we're looking for that specific antigen. And then we have the next type of ELISA, which is where we're looking for what that antibody binds to. Here is a pretty important question. If you have high amounts of IgM in your titer, in your serum sample, why does that suggest a recent infection? So that's something you guys should think about. I'm not going to go over it because it's something we've talked about a lot, but you guys should be able to answer this question of, if we have a recent infection, why are we seeing an increase in IgM? So kind of go through that. If you're aware of that and you kind of have that idea, good. If not, go back through your notes and see why IgM, we would see a high number of IgM in a recent infection. So here's the indirect ELISA. So the first ELISA that we were using was finding if we had antigen in sample. This time we're going to take the serology and we're going to find out the antibodies that the patient has, what that antibody is actually for. So we're going to have a whole bunch of different antigens on a solid sample, and all these antigens are actually labeled to know what type of antigen it is. We're going to run the patient's serum through that solid sample. Whatever antigen it binds to, we'll then wash away the excess. We'll do the same thing, apply a new antigen that can bind to or a new antibody that can bind to this antibody that has a label on it, usually a fluorescent label. And then when we turn on our fluorescence, we'll see which antigen our patient antibody, this orange peachish one, bound to, and it'll let us know what that antibody is for. So you do your preliminary titer and you find out, okay, the patient has a whole bunch of antibody in their serum. Now we need to take that antibody and find out what it is an antibody for. So this is going to give you a little bit more of a specialized diagnosis here to know, all right, the patient just has a whole bunch of antibodies for this. Okay, well then they probably had a recent infection of that. Here, this is just an example that he goes through. It's another case question, patient X. Once again, you don't have to know these too well. This is just to get you all thinking from a different perspective. And he makes that pretty evident that thinking from that perspective is very important, because all of block 2 is going to be this perspective here on cases. They will not be showing you pictures, so you don't have to worry about it. Here we can see some gram-positive diplococci again. So we see it's 2 cocci right next to each other. These are round. These aren't rods. Rods are very, very distinct. And they are purple, so it's showing gram-positive diplococci. So once again, diplococci in pairs. What is used? So indirect ELISA it won't identify IgE levels. You'll use a titer, a serum titer, to detect IgE levels. The indirect ELISA is telling you what that IgE has gone through with variation to be able to bind to. So what specifically that IgE was made for is what the indirect ELISA is going to tell you. It's going to tell you what antigen that IgE was made for, but it's not used to identify IgE. The IgE, to identify if you have high amounts of IgE or IgM, they just take a blood sample, they spin it off, they look at the serum at the top, and then when they look at that, you can see antibodies there. So you can do that, and you can quantify how much IgE or IgM you have in that sample. But the ELISA is used to specifically say, this IgE is for this bacteria or this infection. And then here, just another gram stain, looking at sputum, so spit. Not really spit, but phlegm, coughing up phlegm. Plate cultures, just some other media they use. Once again, he just wants you to have that idea, because as a physician later on, this is the kind of stuff you're going to be looking at. You know, no one's going to tell you, like when you look at this, no one's going to say, hey, this is this. You have to be able to look at this and say, okay, well, if this is a diplococci that is gram-positive, I should be able to administer this antibiotic now. And then as we go through this other testing, I can then narrow it down and change to a more narrow spectrum antibiotic. And then here's that same picture again. So we know it's strep pneumoniae because it's a diplococci. It's really the only diplococci you're going to come in contact with. But then we can also see that capsule. All right. And then these questions, like I said, go through them. He went through them in lecture, so it's important. And then the additional questions from the textbook are very, very helpful as well. All right, so let's get on to the third and final lecture. And then you all can have freedom before Dr. Muffley's lecture. And then I'll see you all again at 3:30. Regarding pathogenicity, islands on the genetics diagnosis, does that mean if there is low? Yes, so if there is low GC binding, that means that there should be high AT binding. So those regions would be high in AT regions, but that's not going to be the differentiating factor that they'll most likely test on is the fact that it's low GC. But that is a good way to think about it, is if we don't have the GC binding, then what else could be there? All right, so pathogenesis, this one's very important. So our objectives, the chapters, like I suggest from now on, if you haven't been reading the chapters, glance through them before, just do it maybe on like a Sunday and kind of read through the chapters for the week. You can just kind of glance through. You don't have to really dig too deep. So our normal flora is just the bacteria or the microorganisms that are living on our body or in our body, and they are there. They don't, they're not pathogenic, but they can cause disease state if the colony numbers are disturbed, and that's referred to as dysbiosis. This is something that if you were there in the lecture on Tuesday, he explained, and it becomes kind of a big deal in block 2, but he will test you on it now. I also highlighted this here because this is pretty important to note that it's not just bacteria in our normal flora. There is yeast, there are viruses, and there are even bacteriophage in our normal flora. There's going to be on our skin and our mucous membranes. So the skin being the exterior, the mucous membranes being our entire GI and GU tracts. So this image is the same thing in the same slide that you see on Tuesday's lecture. You won't have to know these yet, this list, but in block 2, you will have to know all of these genus and a lot of the species involved. So the traits of a pathogen, these are pretty important. I highlighted the key factors here. So what demarks a pathogen is its ability to invade the host, its ability to resist the immune response, its ability to attach to host cells or certain organ systems,
and then gain access to our nutrients and multiply. I referenced this before, and I was talking about that growth curve. So here, this is when it's gaining access to our nutrients and multiplying. This is going to be the log phase. And then also to transfer from one host to another. That's a pretty important feature of a pathogen, is its ability to go from host to host. And then pathogens usually cause disease. Not always, but for the most part, when we're thinking of a pathogen, we're thinking it's causing disease. Some of the entry portals, I'm not going to really go through this so much. You guys should kind of understand this here. This is any area that a pathogen can get through, or any sort of way it can enter our body. So colonization. Colonization is just the growth of that bacteria. So we have two terms that we refer to. Over-colonization, which that term implies that there already is a colony there. With that colony there, yes, this lecture is going to be on the exam, Sophia. This is the last lecture on the exam, lecture 10. Then the lecture on Tuesday is not going to be on the exam. So over-colonization, like I was saying, is implying that there is a colony already there, which means that this is from our normal flora. So over-colonization can only occur in our normal flora because we already have normal flora in these non-sterile environments. So like I said, the GI tract, the GU, so the genitourinary, and the upper respiratory, anything that is non-sterile. Here, we already have bacteria there. If we have dysbiosis, then that bacteria will start to grow out of control because this is just normal ecology. The bacteria there, we have more than one type, or bacteria, virus, any sort of other microbe that we see in that environment. They all compete for resources, so they keep each other in check. When one of those is gone, whether it's through overusing of antibiotics, then we can have an increase in the other type of normal flora that's there. When we have that increase, that's when we get over-colonization. Then colonization is when we're going to have an external pathogen in a sterile environment. So that's the big difference here. Is over-colonization is in a non-sterile. Colonization is in a sterile environment. Our sterile environments being blood, lymph, central nervous system, lower respiratory, and internal organs and tissues. All of these here, if we have colonization in here, then we have an infection of an external pathogen. With over-colonization, we can get it from the internal or the external pathogen. It's mainly going to be an internal normal flora that's going to be overgrowing, which is what you see, and that's what you have when you have when you take antibiotics and you have bad GI symptoms. It's because you have over-colonization of normal flora that aren't being affected by the antibiotic. So kind of know what your sterile and your non-sterile environments are, and which the differentiating factors between over-colonization and colonization. So our modes of transportation, these are pretty important to note. Also, it's important to note the examples. So aerosols just being air droplets. So any sort of mucus or saliva droplet from a sneeze or a cough, anything like that. Our examples of that are going to be influenza and tuberculosis. Here, there's just that image that a lot of science teachers like to show. This is what an actual sneeze looks like. They've just adjusted the background and the camera so that way you can see all the aerosol droplets. You don't normally see all these aerosol droplets, but this is how those aerosols that influenza and tuberculosis can transfer from person to person. Fecal oral should make sense. That's E. coli and Norwalk. Norwalk is the norovirus, so that's the cruise ship virus that you have a lot of people with GI issues on cruise ships have. I think the biggest cases have always been on like Carnival cruises. So make sure you know those two examples of fecal oral. Sexual, so these are sexually transmitted. Gonorrhea and herpes. This is through sexual contact of GU. Arthropod-borne here, I know he explained this one pretty well, Zika and malaria. So this is from a human host, and then it's transferred from human to human through an arthropod vector. So any sort of bug. It doesn't have to be mosquito, but mosquito is the most likely cause. But just make sure you know this is human to a bug to a human. Zoonotic is going to be between animals and then going from animals to human. So it's an animal reservoir. So the microbe or the pathogen is normally occurring in animal species. Then from that animal species, it can then be transferred to a human. Our example of this being rabies. And then other modes of transmission. So zoonotic with an animal reservoir and arthropod vector. So this is a combination of the other two. So we have here the animal reservoir, and then the vector being the arthropod. So here being the flea. So this actual picture is going to be of the plague, the bubonic plague. So the animal reservoir being the rat, and then the flea takes a bite from the rat and goes to the humans and then infects the human with that. Another example being Lyme disease, which our vector for that is a tick. So he kind of mentioned in passing. I would make sure to know that most of the time, like Dr. Anderson would think this is just commonplace information, but make sure you note that Lyme disease comes from ticks and plague comes from the flea as the arthropod vectors. So then some virulence factors, we've talked about these a lot. Bacterial byproducts that can cause damage to the host, and many times it causes disease directly through destruction. And we have some enzymes and some toxins. We're going to go through a lot of different examples of these. Make sure you know these examples and how they work. Very important. So flagella is for motility. It is antigenic. So what it does do is it induces a humoral response. So an antibody-based response is going to be what the flagella target is for. So you can see here, like this is a flagella. It's a lot longer one. This one a little longer. Some of these are pili as well here. So not all of these are flagella. Some other methods of pathogenesis. So we have adhesion, invasion, effectors, exotoxins, evasion of host defense, and then biofilm formation. We're going to go through all of these. All of these are very, very, very important. So adhesion, we've talked about this a little bit, the pili. So pili is going to be our main source of adhesion for bacteria. And we can see they're binding onto glycoproteins on the host cells, so on our cells. So we have pili and then non-fimbrial adhesions, so two different versions. This allows the bacteria to adhere to any sort of cell lining. So whether it be blood vessel, the bladder, the intestine, any other sort of mucosa. So this is how they can avoid that, being excreted by attaching onto those surfaces. So the pili here, you can see the pili is transmembrane. And like Dr. Anderson mentioned, this is gram-negative. We know that because the thin peptidoglycan, the periplasmic space, and the outer and inner membrane. Here the orange just being the LPS, or copper-colored things. So you can see the pili goes throughout the entire membrane. It's not just on the outside. It actually has some interaction with the inside of the cell, and that will become important later. So then some non-fimbrial adhesions. So non-fimbrial are usually found in gram-positive, and this is where we're going to see LTA. So LTA before we said was one of the surface proteins on the outside of gram-positive, so LTA and TA. These are important in adhesion. So this is their function, is actually adhering to host cells. So we see this in Staph aureus and Strep pyogenes. Make sure to know those examples, and also make sure to always correlate LTA with gram-positive. So invasions, this is different than adhesions. So adhesion is just binding to the host cell. Invasion is actually entering into the host cell. So we have two ways. The first one that I'm going to talk about is endocytosis, and this occurs with facultative intracellular and obligate intracellular. So obligate intracellular just being chlamydia and rickettsia, so they have to live inside the host. Facultative intracellular, we've mentioned before, being some other bacteria that can hide within host cells, but they don't need to live in that host cell. And the other way is hijacking the host cytoskeleton. So here, if the bacteria or the microbe will hijack the actin filaments or microtubules that help build the cytoskeleton inside of the host cell, they now not only control that cytoskeleton, but they control that transport system within the cell. So they can transport their byproducts or their toxins from one side of the cell to the other. So it allows for easier motility.
Wait, so just to be clear, invasions are only going to be in facultative intracellular, and obligate intracellular?
No. So we have two ways that the invasions work. One of them is through the second bullet point. That's endocytosis. Mm-hmm. So that's the intracellular bacteria getting inside. And then the other one is an invasion where we're actually, it's gone through adhesion. The bacteria is attached to the host cell, and then it injects proteins to take over the actin filaments and the microtubules inside the host skeleton. So it's either going to invade by invading small pieces and taking over the host cell machinery, or it's going to actually physically invade, and the entire bacteria will be inside the host cell.
Gotcha. Thank you.
No problem. So some examples here, like I said before, phagocytosis or endocytosis, and it's to provide safe harbor. So to allow the bacteria to hide from the immune response. And you should remember these examples. This is examples that Dr. Pross gave in her last lecture, I believe, the very last one she did, where she gave some examples of bacteria that can hide inside host immune cells. And then effectors are a little different. Effectors are going to be anytime the bacteria is going to have something that's going to actually alter the structure or growth of the host. So it can induce or inhibit apoptosis to prevent the host from dying or to kill the host off so that way it can then spread. These are usually associated with invasions because that's how the effector will actually get inside the host cell, is through that first evasion example. So our most important effector example, I know Dr. Anderson mentioned that this was important. He is not joking. There will be a question on this. There might be two, but there will for sure be a question on type 3 bacterial secretion systems on this test. And then in block 2, you're going to have more than one question on it because he will go over it again when you go over E. coli. So type 3 secretion system is a contact-dependent secretion system. So that means is the gram-negative bacteria, because it's only in gram-negative, has to physically interact with the host. And then once it physically interacts, it can secrete something that will allow it to then impart its own effect onto it. So it is spanning both membranes, and it does create a fusion between the eukaryotic cell and the injection pore. So we're going to get to that. We're actually going to talk about that, and I know Dr. Anderson did too. So what's actually occurring in this is the type 3 secretion system. Once you have that contact-dependent interaction, then the bacteria, so the gram-negative bacteria, will inject a toxin into the host cell. That toxin will elicit a specific response and allow that bacterial cell to infect more. This is done mainly in EPEC, so a strain of E. coli that you're going to go over in block 2. But make sure you know EPEC now, make sure you know what's occurring in this secretion system and the factors in it. So here it is. Here's the actual type 3 secretion system. Remember, it is contact dependent. So we have our E. coli here, our EPEC. It binds with adhesion factors onto the host cell. So this would be an intestinal cell right here, because you can see it's got the little the villi. So that host cell binds with the EPEC. Once that happens, then we create our injection pili. So this is an injection pili. So this is where the E. coli can actually send effectors from itself into the host cell. What it's going to send is going to send this TIR. This TIR is a receptor. It's actually a receptor for E. coli itself. So what E. coli is doing is it attached here through adhesion. It's sending receptors onto the host cell. This receptor will then be represented on the outside of the host cell, and it allows E. coli to attach even further. Everyone see that? So we have the initial contact here. This initial contact allows to create this tube that we then send the TIR effector. The TIR goes from the E. coli into the host cell, from inside the host cell as receptors on the outside, then binding other moieties on the outside of the E. coli to give an increasing interaction. Then from this increasing interaction, then the E. coli can then go through and destroy these actual host cells. So this TIR is allowing it to interact with the host cell more to then allow the E. coli to go through its process and actually destroy the host cell. So make sure you understand that process. Make sure you know it's the type 3 secretion. It occurs with EPEC. It's contact dependent, and that EPEC is sending TIR as a receptor. So the exotoxins, this one's also very important. Here you need to memorize these toxins. So exotoxins are going to be released from bacterial cell. We are going to talk about the difference between exo and endotoxin. There is a staunch difference. So an exotoxin is released, and it can act on the host cell membrane, it can act on protein synthesis, and it can act as a neurotoxin. We're going to talk about examples of all of these, and there are A and B subunits that you do need to know. So toxins as a whole, they just harm tissues. We have the endo and exotoxins. We've talked about endotoxins before, LPS being our endotoxin that we've talked about on gram negatives. I'm not going to go over this because we just went over it earlier this morning. So our exotoxins, like we said, are excreted. So endotoxins are part of the cell membrane. They don't leave the cell. An exotoxin is excreted by the microbe, and it's excreted onto the host cell to have a specific response. So they have A and B subunits. The A is usually the active subunit, and B is the binding subunit. So those are good ways to remember that. We can see that in both gram-positive and gram-negative. We're going to go over some examples. You need to know these examples. You need to know them. You need to know which subunit is involved, and you need to know what it does. So if you can commit anything to memory, commit this slide 33 to memory. So the first one, Clostridium perfringens, it uses an alpha toxin. And what the alpha toxin actually does is it lyses the cell membrane. It's So you can see right here, it actually causes lysis of the cell membrane. If we lyse the cell membrane, it can cause death. So make sure you note that. Staph aureus, it has an alpha toxin as well, but the alpha toxin does something different. Here, the alpha toxin just creates a pore in the membrane. So by creating a pore in the membrane, then we get leakage of internal contents of the cell, which then causes cell death. So both alpha toxins here cause cell death, but they do it in completely different ways. So make sure you know these different ways. So the alpha toxin in perfringens causes lysis of the membrane. Alpha toxin in aureus causes a pore formation. Then we have diphtheria toxin. Diphtheria is both A and B subunits. And what it does is once it gets incorporated into the cell, it prevents protein synthesis. So it inhibits our cells from actually creating proteins. This will then lead to cell death in our cells. Then cholera toxin, it is also A and B. It has both A and B subunits. Here, what this one does is the A subunit interacts with CAMP. By increasing CAMP, it forces our cells to export more or to efflux more of our actual nutrients, so of our electrolytes. So by increasing CAMP, we are forcing more of our electrolytes out of our cell and into the interstitial area, which is going to be, this is occurring in the intestines. So our intestinal cells are releasing more sodium, chloride, bicarb, potassium. They're getting rid of all those electrolytes. As they do that, water will follow. So it's not actively pushing out water. It's just increasing CAMP to increase electrolytes. When it increases the electrolytes, the water follows, which then causes diarrhea. This is going to cause diarrhea referred to as rice water stools because we're just dehydrating the body by pushing all of those ions out, and water just passively follows those ions. Then we are dehydrating all of these cells. So that's how the cholera toxin works. The last two, these are the neurotoxins. So tetanus toxin, what it actually does is it inhibits the inhibitory transmitter. So by inhibiting an inhibitor, you get increase in stimulation. So tetanus toxin is just going to increase stimulation. So you get overstimulation of neurons. With botulinum toxin, it's actually going to inhibit acetylcholine. So by blocking acetylcholine, it's blocking a stimulator. So if it's blocking a stimulator, that means nothing is occurring. So here in botulinum toxin, we get paralysis because we have nothing occurring, no stimulation. In tetanus, we get essentially a paralysis. It's a tetanospasm, but we get a paralysis because of overactivation. So here, the muscles are clenching and over-clenching too much. Here we get, in botulinum, we get a flaccid paralysis where everything just relaxes and there's no stimulation. So make sure you know those differences. I cannot stress enough how much you need to know these 6 exotoxins because there were multiple questions on these 6 exotoxins. And then they come up again in block 2. So you need to know it now for this test and for the next test.
Daniel?
Yes.
Do you mind going through diphtheria one more time?
Sure. So diphtheria has both an A and B subunit. So as we said before, the B subunit is the binding one. So you can see in the image here. See if I can. Bam. So the A and B subunit, the B subunit binds. Once it binds, we have endocytosis. The A subunit is then the active one. What the A subunit is, it inhibits protein synthesis. So by inhibiting protein synthesis, the toxin itself induces cell death in those proteins or in those cells that it's actually infected. So it just releases the toxin gets in, the A subunit prevents protein synthesis, and that will result in cell death.
All right, thank you, Ben.
No problem. All right, so then evasions, we've gone through some ideas of evasion, and then actually some of these were already expressed by Dr. Pross in previous lectures. So we have the capsule. The capsule helps the bacterium evade phagocytosis. Then we also have some other ones like protein A on Staph aureus. This was mentioned before. Protein A actually prevents Ig binding. So not just IgE, but any Ig binding from onto Staph aureus. So that prevents an immune response. So that's how it can evade the immune response. Then we have 2 examples for gonorrhea. So our first one being an IgA1 protease and then antigenic variation. So IgA1 protease is a protease that actually breaks down IgA. So if it breaks down IgA, that's breaking down our antibody or our immune globulin in the mucosal area. And we should know that gonorrhea does infect the mucosal area, considering it's an STD, and the GU is referred to as mucosal. So by gonorrhea actually breaking down IgA, it's destroying the only immune globulin in that mucosal area. So now it doesn't have to worry about any sort of immune globulin or antibody in that mucosal environment. It can completely evade or destroy the immune system there. Antigenic variation is a little different. Antigenic variation is where gonorrhea can actually change the genetic expression of its pili. So what it does is, I want to go through it in an actual picture a few slides later on, where there are certain genes presented on a pili, and gonorrhea has multiple different variants of those genes. So if you create IgE or an IgA specific, mainly an IgE specific for gonorrhea, because your host, your immune system is activating, when you do, you're creating it to a specific variation on that pili. Once you create that IgE that has high affinity for that pili, gonorrhea will then go through, internalize that pili, and put out another one with a different variation. So now that IgE that you made is completely useless because it can't bind that new pili with that new genetic variation. And we'll go through it again on a picture. So capsules, we've talked about this a lot. We've mentioned a lot. You've seen it. This is like the third or fourth time they've talked about strep pneumoniae in a capsule. I would make sure to remember that. I mean, if they've said it 3 times, they obviously want you to know this is the example of the capsule. So do not forget that specific example. And then here's the image for variation. So we have our 3 different variations here. And we can see that this is just the mRNA as it goes through and creates that pili receptor on the outside. First, if it has that beta form and you create an IgE for that beta, gonorrhea will then internalize that beta and then put on an alpha. And then once you create IgE to that alpha, it'll internalize and then create a different one. And then by the time it's gone through all of these, when it gets back to its variation of beta, your antibodies to beta have died off, because remember, antibodies don't last for that long. So then it'll do it again. It'll keep doing it. And this is why it's so hard to treat gonorrhea. And this is one of the reasons why we can't eradicate gonorrhea once you have it. And then our last one, biofilm formation. So I kind of talked about this in our Friday review last week. If you were there or listening, I gave the kind of ridiculous example of March of the Penguins, but I'm gonna do it again. So the biofilm is just a structure, organized community of bacteria. So what it is it's a whole bunch of bacteria that are stuck at an interface between a solid and a fluid. And when they get stuck there, they then create this whole community. And this whole community essentially protects. It's the bacteria protecting each other from any sort of harm. So they're protecting them from the immune response, and they were protecting them from antibiotics. The way they do that is very similar to how the penguins did it in the South Pole when they stand there and they're all freezing. So if you have all these penguins in one group, and you have these terrible, terrible freezing winds going around, causing damage and injuring the outside penguins, the outside penguins will then come into the inside will switch. So we'll constantly be switching around the inside and outside so that the penguins are getting slight damage, but not enough to actually kill them. This is the same idea behind this biofilm. So we have this group of all these bacteria. The ones on the inside are completely protected. The ones on the outside are exposed. As damage occurs, the ones on the outside will come back in, and they'll switch place with the ones on the inside. So they'll constantly be rotating so that no bacteria in this biofilm is getting enough damage to cause death. And if we do that, we're constantly protecting the bacteria here. This is very different than planktonic growth. Planktonic growth is just free swimming growth. So this is growth within one specific medium, whether it be an entirely liquid medium. So this is what you see in sepsis, when we just have a whole bunch of bacteria growing, freely swimming around within the blood. You can also see this in lymph fluid. You can see this anywhere where it's just free swimming. The biofilm only occurs at that interface. And you'll learn why later on in physiology, why it's only occurring at that interface and why some bacteria get stuck onto that interface. So here we can see the actual diagram of what's occurring. So we have free-floating planktonic cells. As they're floating in this fluid, some of them will get stuck because of turbulent flow in the fluid, will get stuck onto that hard surface, that solid surface. So whether it be the inside of a blood vessel, whether it be on a catheter, anything that is firm that is exposed to that constant motion of the fluid. Those planktonic cells will get stuck and then become sessile. Sessile just means they're not moving. Once they're there, they'll get stuck, they'll start multiplying, you'll see more. They will also start interacting with each other through quorum sensing. As they interact with each other, they will also recruit other planktonic cells. And now they create this entire mass, just like the penguins in the South Pole. So we can see here, they're here, but they're not stuck there. They're constantly moving around in this huge community and organization to protect them and prevent any sort of infection or any sort of antibiotic or any sort of immune response. So here we can see some examples of it in gum disease between the saliva and your teeth. where you can see the biofilm. You can also see it in catheters. This is one of the more common areas that you see it in. And then any sort of implant, because we'll see blood flow going across any sort of foreign device that is a hard surface that wasn't already designed by the body. Here's an example. Anytime they give a very specific example, I would make sure to know that example. So Pseudomonas aeruginosa, this is a bacteria that can create this biofilm within the lung of cystic fibrosis patients. This is pretty important because already cystic fibrosis leads you to higher chances of infections. And then once you get this biofilm formation from Pseudomonas aeruginosa, which, by the way, this bacteria is found throughout all of the soil in the United States, in Florida for sure, some colder environments, not as much, but this is found in soil. Once you get that biofilm, it's very, very hard to treat because antibiotics won't work. Your immune system will not respond to it. So it's very, very hard to even consider treating this biofilm. So we can see that we have here another image of this biofilm, this group of cells, and they have this matrix that they're developing through quorum sensing on the outside. And then we have this planktonic cells that then will leave off and go and infect another area. So it's just creating a nice little hub here for this bacteria to avoid any sort of interaction. Just an image of what the biofilm looks like. So you can see all the sessile cells, and this is going to be that that capsule that they create on the outside, that actual physical film. And then here are some questions. Again, I would always suggest doing those questions so that way you guys can have a little bit more practice. I would also suggest going back through doing the practice test if you haven't done it already. When you go through the practice test, like always, don't just find the right answer, go through the wrong answers and explain why they are wrong. That's very important because that can help you learn multiple concepts. And then go through the poll everywhere questions. Those are also very important to go through. All of those, like I always say, have been approved by Dr. Anderson, Dr. Pross, so they're indicative of what test questions could look like. They're not the same as the test questions because we made sure to make sure that they were different, but they are very indicative of what test questions could be like. So that is my review. I do not have any practice questions today because you have your practice tests, and I want you guys to focus on your practice tests. Does anyone have any questions before we sign off? Okay. Here we go. So for the last example, the secretion of exopolysaccharides allowed to invade or interact and stay in the biofilm. So the exopolysaccharide, that's actually the actual physical film that these bacteria create that can give them kind of like a shelter and protect. So that's something that they will produce later on, and that's the actual physical film that can prevent antibiotics from from actually directing their attack. So that's what the exopolysaccharide does. Any other questions? If you have a question, you can either type it in the chat or you can come on and speak in the Teams.
Did you go over the botulinum toxin? I'm a little confused on whether it's the actual A and B toxin.
So with tetanus and botulinum, we don't refer to them as A and B because they are neurotoxins. So the only ones we're concerned about with A and B are going to be C. perfringens and Staph aureus, both of those being the A toxin, and then diphtheria and cholera having the A and B. So with tetanus and botulinum, we're not really too concerned whether it's A or B. What we're more concerned with is it is a neurotransmitter toxin. Does that answer your question?
Yes, it does. Thank you so much.
No problem. All right. Will you hold TA office hours on Friday morning for last minute questions? So no. As I said at the beginning of this review, and I put out in the announcement, my TA office hours are going to be following the Q&A today. So the Q&A today that Dr. Anderson, Dr. Pross are having, I will be there in class on the Teams meeting. They're going to do their questions, and then after they do their questions, I will then follow in, and I will hold my office hours from the moment they are done, probably for 2 hours. It depends on attendance, and it depends on how long they take. But I will have just open office hours at that point. I will not hold open office hours Friday morning, but I am always available through email. So if you don't ask the question today, and it's something you come up with later on or over the night, or Friday morning, just send me an email. I try to get back to people as soon as possible. Sometimes if you send it to me at like 3 or 4 in the morning, I will not be awake, but I'll respond right when I wake up. So I won't do it tomorrow, but today I'll have the open office hours. That is a fantastic question. I don't know if the Q&A is only online or in person as well, because I know there was an issue before of a potential for a COVID case, but I know that was it was a false alarm, which thank goodness for that person. So I'm not too sure. I would send Dr. Anderson an email or send Rose an email and ask them if it is in person. I will be doing it online. I will not be in person. I will be online. So I'm not too sure if Dr. Anderson and Dr. Pross will be in person or just online, but you can send them an email and find out. Does anyone else? So Alexis is saying they sent an email and said it is online. I do know they sent that before. I just don't know if it lasted through. So we've got multiple people. Like I said, I'll be online. Online is always a safe bet. You're not going to miss too much in this case for doing it just online, because what they're going to be doing is just having questions for you all. Does anyone have any other questions?
I do. How in depth do we need to know the alternative complementary system and the complementary system?
So for those, like I've mentioned before, I would make sure to note what elicits both system. So the classical is activated by antibodies and the alternative is not. And then I would know the main factor between them and also what they do. So the main factor being C3, and then what they do, like their actual function, how they activate a response.
Okay, thank you.
No problem. Any other questions?
Yeah, Daniel, yeah, so I was wondering with negative and positive selection, are those dealing with both self-MHC and self-antigens? I wasn't sure if it was like one or the other.
So negative and positive selection is when we're doing the education for the T and B cells. And what it's doing is it's seeing how the T or B cells respond to presentation of MHC. So the Tregs will present MHC. It doesn't necessarily have to be MHC class 1 or 2. They're just presenting MHC as a whole. So it could be 1, could be 2. It doesn't matter too much. But they're presenting MHC and they're seeing how the cells react to MHC. So in negative selection, that means the T cell in question, is overreacting to the MHC. It's overstimulating. So then we negatively select, we get rid of it. For positive selection, that means it's reacting to a certain point that we need it to react to MHC. That makes sense?
Yeah, that makes sense. I was just getting confused about like if part of that was like the presentation of the self antigens as well, or if it was just the MHC specifically.
So the self antigen is MHC. I know that was confusing because for like the first 3 lectures, she just threw it out there as like, it presents self antigen. And then in one lecture, she's like, oh, by the way, the self antigen is MHC. So the MHC is actually the self antigen. That is what they use for presentation. That is the only self antigen that is used.
Oh, okay. So there's like nothing being presented within that MHC like a normal antigen would be presented. It's just
no.
It's the antigen. Okay.
Yeah. So what they're doing is it's the Treg presents the MHC, and then it sees, will the T cell bind? So if the T cell binds to the MHC, fantastic. If the T cell binds to the MHC and then starts eliciting a response, now we have to negatively select. If we present the MHC and the T cell doesn't even bind, that's anergy. And then that one we have to negatively select for too, because it's not responding at all.
Perfect. Thank you so much.
No problem. Any other questions? People are dropping like flies. All right. So if there's no other questions now, I will head out, take my break before I see you all at 3:30. Good luck with your studying, like always. If you have any email or if you have any questions, just email me. Try to email back quickly as possible. And good luck. Make sure to get rest. Do not pull an all-nighter because you will forget. There's a lot of concepts to worry about here. If you pull an all-nighter, you will forget a lot. So try to get a good amount of sleep. Wake up early. Give yourself plenty of time. Also, remember the test is at 2 tomorrow. And after the test, we will not have a TA review. I think Justin is considering doing an anatomy review. that would be a good thing to get on if you can, because you do not want to get behind in anatomy, as you guys probably saw with the anatomy test. Good luck, anyone. If anyone has any questions, just email me. Have a good day.